I think it's still shocking to people when they learn that this disease is still the biggest killer of kids in the country. It still kills more people under 40 than most other diseases. The rarity of the cancer right now shouldn't determine the overall investment. We've seen really significant improvement in survival outcomes for other cancers, long-term survival outcomes. The reality is right now you are much more likely to survive other cancers than you are brain cancer, and brain cancer is one of the worst cancers you can get in terms of your survival outcome over a 5-year period. The reality is also that there's a lower percentage of people that get this cancer and therefore lower attention and less ambassadors for the cause. And I think now's the time, and building on the momentum of the Brain Cancer Centre, we've got a critical mass of people who are right now at the cusp of being able to take discoveries into the clinic, to build the pipeline of researchers who are going to come in and have a career in brain cancer. Now, a young person coming in to have a career in brain cancer as a PhD and having the security of their career over the next 30 or 40 years, the outcomes are incalculable. The inspiration that I take out of the Brain Cancer Centre every day is both through patients who have the disease, but also through incredible researchers, the likes of which we've been able to connect within the Brain Cancer Centre. What I would love people to understand is to have the smallest insight into their world and what it takes to deliver the outcomes, how incredible the detail is, how much understanding we have and that we don't have, and how the work happens within the laboratory environment that takes us towards those drugs and therapies that we need to create a better outcome for patients.
I'm Dr. Sarah Best and I co-lead the Brain Cancer Research Lab in the Brain Cancer Centre, which is physically located at the WEHI in the Personalised Oncology Division. And within this division, the brain cancer researchers work closely with researchers working on lung cancer and pancreatic cancer, where we can really harness all the different tools that everyone uses to improve research overall. This is where the Brain Cancer Research Lab does all of their work. So the surgery happens just right next door at the Royal Melbourne Hospital, and then we have the material delivered straight into our cold room so that we can immediately start working on that material while it's still alive, because it doesn't actually last very long outside the patient. But once we can start researching on it and turning it into these models, we can investigate it for many different years and grow the tissue up and do lots of different types of research on it. We also have a cold room, which is really important in the lab because a lot of things that we do have to stay really cold. And so there are experiments that we need to perform at 4 degrees Celsius, and these experiments we can actually perform in this room. Something that you might not realise about labs is that we need a lot of freezers. Everything that we have needs to be frozen, both at -20 degrees, which are these silver freezers, but mostly at -80 degrees, because a lot of the different RNA and proteins that are found in the cell can degrade. So we really need to snap freeze and freeze really well the cells in between experiments. So freezers are really important. They're also ridiculously expensive. These boxes here are incubators, and because the cells and tumour cells that have come from patients, our body temperature is between 35 to 37 degrees Celsius, so we also need to keep the cells that we propagate at that temperature as well. So this is a cell line that our lab's generated from a patient sample, and you can see the red solution in there, and that's the media. It has a bit of a red colour to it. You won't be able to see the cells. For that, we'll need to look at a microscope. So that's a growing neurosphere from a patient sample, and that we can grow and expand to perform hundreds of experiments on. We can't really see anything just by looking at it, but we'll be able to see every single cell when we look at this slide down the microscope. Right now, we're looking at about 40 times magnification compared to when we just looked at the slide by itself. But what we can do is look closer. So now we can see the actual architecture of the tumour. Each of these dark purple circles is the nucleus of a cell, and the nucleus is where all that DNA, all the genetic information is kept, the encyclopedia of each cell. And now you can really appreciate these brown regions. And so this positive control tissue is telling us that we would expect to see a lot of dividing cells in this area, so that we can use this positive control to know that our experiment has worked, and then we can look at our brain tissue to see what the extent of staining is. For brain cancer treatment, because treatment hasn't changed in over 2 decades, unfortunately, identifying different factors like this in the clinic doesn't have an impact on the type of treatment patients will receive. But we hope to change that. Since I was quite young, all of my grandparents had passed away from different types of cancer, and I was really interested in biology at high school. And so that really drives me to better understand cancer and brain cancer, and then that all leads to identifying improved treatment for patients with brain cancer. So just being able to equip a lab so that we can perform our experiments is extremely expensive, but then also performing the experiments themselves. And so funding is always a great hurdle when we're thinking about research, and the Brain Cancer Centre and Carrie's Beanies for Brain Cancer has really allowed us to move a lot of our research forward, but we need more funding to keep it going. With a large budget, we can hire more scientists and train up more researchers to work on brain cancer. There are a lot of really advanced experimental tools that are out that are very expensive at the moment. And I'll come back to heterogeneity because we can use these very expensive experiments to really understand at a molecular level the heterogeneity of tumours. And that's an approach that we're working towards in the lab, but it's extremely expensive. And so if we had more money, we could analyse more tumours and get a better idea of what's going on. When we're constrained by budget, we can't employ all the scientists that we need to do the work effectively, and we can only look at a smaller sample size, which doesn't capture the diverse array of patient tumours. I'm really excited about what the future holds for brain cancer research. Seeing the Brain Cancer Centre develop over the last 1.5 years has been so incredible. The number of researchers we've been able to pull together, additional scientific collaborations, and really getting lots of different people who work in different fields in the same room, which has been really important. And what we've seen in this short amount of time just really excites me, and I can't wait to see what happens in the future.
There's absolutely no doubt that we can solve this. Solving this is not actually the question. Will we solve it, will we not solve it? That's not the question. We will absolutely solve it. This is about two things. This is about talent, and this is about resources or money. And if you have enough of those two things, you will absolutely create discoveries that take you towards a cure.